Bienvenue tout le monde. Merci d'être venu. It's my pleasure to introduce Dr. Jennifer Ronholm, who's a relatively new professor at McGill in the Department of Food Sciences and Agricultural Chemistry. Yes,
yeah.
So that's the full, Food Sciences since January 2017, so almost a year. Jennifer did her bachelor's degree at the University of Waterloo and a PhD at the University of Ottawa, identifying virulent strains of Listeria and Salmonella. She then did a postdoc at the Canadian Astrobiology training program, looking at how microbes survive in the high Arctic, followed by a visiting fellowship at Health Canada in Ottawa, also doing more food safety and then bringing genomics into food safety. And then, as I said, it's been at McGill, I'm going to point the wrong, McDonald campus for almost a year now. So very happy to have, I don't know, yeah, somewhere. So we're very happy to have her in the greater Montreal area, and today she's going to talk to us about interactions between bacterial pathogens and microbes.
So thank you for having me. I was excited to come and give the talk today, but like Jesse said, I came in January, so I don't have too many results yet. So most of what I talk about between interactions between bacterial pathogens and the microbiome is theoretical and ideas. So I am going to talk about some work from my previous two postdocs that kind of got me to the ideas we're on now. So I'm calling this McGill round one because I did my first postdoc and now I'm back. So this was, I'm going to give a quick overview of what I did during that first postdoc. So some of it was kind of interesting, but not related to food. So one of the big projects I had first round at McGill was microbiomes in the high Arctic. So what bugs live up there and what are they doing? And this has a lot of relevance for two reasons. And one is climate change and the other is astrobiology. So in terms of climate change, there's a lot of concern that when everything starts to heat up, as we're seeing it do now, that the Arctic is going to heat up more than anywhere else. And this warmness is going to lead to a wetter Arctic. And that's because the permafrost is going to melt and the waters are going to rise and all the apocalyptic stuff. But the big concern there is that right now the Arctic soils are actually probably eating methane. And that's actually really good for us because it takes methane out of the atmosphere and turns it into carbon dioxide. And methane is a lot stronger greenhouse gas than carbon dioxide is. So there's a lot of debate at the time, this was a couple years ago now, is this a chemical process or is it microbiological? Like it really looks like these soils are eating methane, but why and how? So the work we did was actually measuring methane soil flux in the Arctic. And you'll see here, these are called polygons, and they're actually formed by these repeated furious freeze-thaw cycles. So all the water will go down into these troughs and then freeze in the winter and make the troughs wider and deeper. And you can actually see that when we were up there, our airstrip kept getting these big troughs in them. So we'd spend one day filling in the troughs with soil so the airplane could land and take us home. But the other thing is that those are getting deeper and wetter, and they're kind of eating methane at a different rate than the rest of the soil. So it's nice because they might give you a look into what's going to happen as the Arctic warms and wets. So you can see here we're measuring methane flux both in the wetter trough areas and up on the dry land in the middle of the trough. And what we saw here is that these are actually putting out methane, while the inside of the polygon was actually eating methane. So that could be what's going to happen when the Arctic gets warmer and wetter. But we wanted to look at that from kind of a microbial perspective. So we actually did metatranscriptomics, and we were looking for the genes that would be associated with methanotropy, so eating the methane. And we did actually see that, but the important genes, the PMOC, are right here, but we only saw it in the trough. And that kind of, we didn't know what was going on there. But my best guess is that they're producing methane in these troughs, but they're actually eating it at a slower rate than they're producing it, whereas they're not actually eating it in the polygon country yet. So that still kind of leaves it up in the air as to what's going to happen in the Arctic, but it could be a bad thing if the Arctic starts producing methane instead of eating it. And then another project we had up here, we're looking at these saline seeps. So near Gypsum Hill in the Arctic, there's these saline springs, and they're super cold. Although for some of the year they're actually below zero, and they're able to do that because they're 15% salinity. So they're a really harsh place for a bacteria to live, sub-zero, 15% salinity, but they run all year, and you do actually get microbial life in them. So one of the questions we were trying to answer is, what is the actual source of these waters? And the geologists are looking at it from a different perspective. But when we did the microbiome of it, we're actually seeing a lot of marine bacteria. So a possible source is the ocean. And in addition, there looks to be some saline seeps like that on Mars. So people are kind of interested in them from that perspective. So another project that we did, and this one was done at Jeffrey Mine in Quebec, which is in asbestos, Quebec, it's one of the biggest producers of asbestos, was actually looking at the microbiomes that live in and on the asbestos, which you wouldn't think a lot of stuff lives there, but there's actually some neat communities. And what we did in this study was compare the bacterial population living in the actual serpentinite asbestos versus the bacteria living in the quartz that kind of speckles its way through the mine. So you can see here, this is actually the asbestos, and it's very fibrous and kind of like wool, and that's why it pulls apart and makes fire barriers so nicely. And these are pictures of the quartz rocks, but this nice little ring here is actually bacterial endolith living in that rock. So we compared those two populations on a number of different parameters, and it's kind of neat. So you can see in the pictures, this is the quartz rocks, and you actually get a nice strand of asbestos running through there. You can see some fungal hyphae, and you can see the bacterial cells nestled nicely into the crevices of the rock, which was kind of neat. But we also looked at where these guys could actually have heterotropic metabolism, and both in the quartz and the serpentinite, you had a lot going on at plus 20, because lots of stuff happens at plus 20. But when you put the temperature down to -5, you can actually see that the serpentinite community, so the bugs living actually on the asbestos, they're still doing stuff, which is actually neat because there's some big asbestos deposits on Mars. So it's relevant.
Is that a control?
Yes, that is the control. So it's kind of a neat study. The bacterial communities were kind of neat there too. So we looked at bacteria, fungi, and photoautotrophs. And we basically found that there were two separate communities in the asbestos versus in the quartz for the bacteria and the photoautotrophs. But the fungi, there wasn't many differences between the communities in both of those locations. And my thinking there is that the bacteria are probably more specialized. And when we actually looked at some of the sequences in the NCBI database, they did actually match to quartz found around the world. So are they that specific that these bugs are, quartz dwellers? And that's an interesting question because that's what was pulled out of the.
Excuse me.
Yeah.
On the bacteria you use, what's the archaebacteria? Or
That's a good question. The primers we've had, I think, do include that. So yeah, that was the asbestos and quartz paper. And that's currently submitted. So after we did this work, I went on to Health Canada, and I was telling Jesse when I interviewed for my Health Canada position, the question was, do you think that we're going to switch to whole genome sequencing for outbreaks and for the stuff we do as a regulatory agency? And I'm like, you haven't switched yet. So my job at Health Canada was kind of coming up with ways that they could do this and helping bridge that gap, which kind of, it's interesting because it feels to me like they should have had a team of bioinformaticians and a team of bacterial geneticists, and they had a postdoc. But I think they're kind of going that way now, and they're starting, but they're a lot further behind than you'd think they'd be at this point. So their question basically is, can we use whole genome sequencing for outbreak delineation? That's what Health Canada is interested in. And when you're talking about outbreak delineation, basically there's one question. If you have two bacteria, one from a sick person, one from a plant, are they the same? And it seems like an easy question to answer, but when you have litigation issues and, big world out there, there's a lot of different things. It's not that easy of a question. So one of their big questions right now is if we have two isolates and we compare them based on single nucleotide polymorphism, SNP differences, how many SNPs are enough SNPs to say it's a different isolate? And it's actually it's a complicated question, because if you start comparing them between outbreaks, well, what are you calling the SNPs based on? Are you doing it on your core genome? Are you doing it on the pangenome? Are you doing it on phage region? And the answer is very different. There was actually an outbreak in the States where some guy got sick 12 years ago and some guy got sick now. They went back to the factory. That factory had been contaminated with listeria for 12 years. Within the core genome, there were two SNPs between the one from 12 years ago and the one from now. But if you looked at the whole genome in the phage region, there were something like 3,000 SNPs.
None of us work on large scale consistent problems.
Okay.
Can you?
Single nucleotide polymorphism. So you compare two genomes, and they're the base pairs of differences. But how you compare them is, everyone kind of uses a different method.
Technique?
Yeah.
Is it a function of how many times we can kind of block your it must be a movable window where you locate it.
So it's based on the whole genome. So for bacteria right now, no one closes genomes that often anymore. So you have your draft genome, and you can align that either to a core genome, or you can align it to a whole genome, like a closed genome that someone's known, and just call the differences, basically. But yeah, they're working on what the best protocol is for that right now. Yeah, exactly. So we undertook, and this is a really simple experiment to establish a baseline. We took 16 strains of 4 different foodborne pathogens and grew them all up 100 times over to see basically how much it would change over these 100 days. That would basically span any outbreak that you're interested in looking at, with the exception of a couple of listeria outbreaks that last for years. And when we use so this is I'm not going to go through these too much, but when we use the protocol that the Public Health Agency of Canada used, we get one answer. And the Public Health Agencies is basically their pipeline takes your draft genome, aligns it to a closed genome, and calls the SNP differences. So the problem with that, if there is a problem, is that if I have 2 strains, one from the factory and one from the sick guy, you're not comparing these. You're comparing them both to a reference. And that could be complicated, and you might not get all the information. We wrote a separate pipeline that actually takes the 2 strains and compares them to each other based on the core genome of these 2 strains, but we haven't got the pipeline fully working yet. What we did have gave us slightly different results than the PHAC pipeline, though. So the reference guided using the PHAC pipeline, after 100 days of subculturing, the most we found was 4 SNP differences, which is actually a pretty good start at saying how many SNPs is a different thing. How many SNPs makes it a different bacteria? Probably in and around 4 or 5 for most bacterial pathogens. There will be some exceptions, though, if they have mutations in DNA polymerase or proofreading abilities. But when we came over to our de novo assembly, we were up in the 350 single nucleotide polymorphism range, as opposed to being down in the 4. So the point I'm trying to make here is it really is going to depend on the pipeline people are using and the methodology and the philosophy behind how you're comparing these genomes. So also when I went to Health Canada, Vibrio parahemolyticus was kind of the thing I was working on.
It was in.
Pardon?
It was in.
It was in?
Yeah.
Okay. So it's naturally occurring in mostly marine environments. Not all of them, very few of them actually cause illness, but some have picked up different virulence factors that actually do allow it to cause illness. And the mechanism of disease they're working on it, but it's not quite fully understood, and I'll tell you about why in a second. So the kind of central dogma around Vibrio parahemolyticus is that there's two toxins that are essential virulence factors, thermostable direct hemolysin and thermostable direct related hemolysin, TDH and TRH. And these guys poke holes in eukaryotic cells. They do that in your gut and you get sick. The basic understanding is either one of them on their own will be enough to cause illness, or both of them together will be enough to cause illness, and different strains have different combinations. Go to the next slide. So there's a common Vibrio ancestor between Vibrio parahemolyticus and Vibrio cholera, and you actually get a lot of genomic movement between all Vibrio, but type 3 secretion systems come on the same pathogenicity island as the TDH and TRH, so they kind of move around together. And you can see that, so Vibrio cholera has the CTX toxin and causes illness by that, but it can also pick up the virulence factors from Vibrio parahemolyticus occasionally, and that can cause illness too, but when it picks up the virulence factors from Vibrio parahemolyticus, it actually causes a Vibrio parahemolyticus-like illness, not the Vibrio cholera-like illness. And you can see the Vibrio, the pathogenic parahemolyticus ones are the ones that have TDH and TRH. Now there's also non-pathogenic that do not have that type 3 secretion system 2, or TDH and TRH. But when I got to Health Canada, there was a number of outbreaks in recent time caused by this group, which was traditionally understood to be a non-pathogenic subtype. And the FDA was seeing the same thing at the same time. So what we did was we got 4 of those isolates and sequenced them. And we sequenced a bunch more traditionally pathogenic ones and a bunch of environmental ones that didn't have TDH and TRH either. So we made a tree, and the first thing that we saw was the ones that have no well-known virulence factors are in green, and they're not in the same lineage. So we were kind of thinking that maybe they picked up the same virulence factor a couple different times, or maybe they have different ways of causing illness. And the other thing we did was compare the pangenome of the environmental isolates we sequenced to the clinical isolates we sequenced. And so these are the number of genes versus the number of genomes. And you'll see that the clinical genomes are actually fairly well conserved. They don't have these huge expanding pangenomes, whereas the environmental ones have an actual very large pangenome. So the total genes that you find in that cluster. And that actually makes sense when you think about it, because the clinical ones are being selected for on a really stringent criteria. Can it cause illness in humans? Whereas the environmental ones are out there doing any number of things. That makes sense.
Are the environmental and clinical definitions the same as the red and purple, or are those sort of two different?
The environmental ones, none of them had TDH and TRH, so they'd all be considered green.
Okay, so that is how you're defining those.
Yeah. And the environmental ones were acquired from the environment as opposed to the clinical ones, which were all hospital isolates. They had actually been taken from a sick person.
So right, so those labels over are the TDH, TRH negative is exactly synonymous with environmental. Like those groups are exactly overlapping. You know what? Like
no.
There's no clinic. None of your clinical isolates are also negative.
Negative.
Right.
Yeah, 4 of them are.
But they okay.
4 of them are. Yeah. So those are the 4 interested ones that we were really trying to figure out what was there. So we actually did. We compared all the genes and all of them from the TDH, RH environmental and TDH, TRH negative clinical. There were the 862 genes that were in those clinical ones, which was something to look for. And we kind of scoured those in every way we could think of. We looked for any of the virulence factors from any of the other Vibrio. We looked for any of the virulence factors from other gram-negative pathogens, and we started looking for anything that looked like a virulence factors, and nothing was hugely jumping out at us. But then we came across, those 862 genes, type 6 secretion systems. So these ones that were causing illness seem to have this extra type 6 secretion system that you only normally see in clinical isolates. And one of them, type 6 secretion systems, fairly recently they were figured out. They're a way for bacterial, mostly gram-negative pathogens, to fight with each other. So they do other things sometimes in terms of eukaryotic interaction, but for the most part, they control antagonistic interactions between gram-negative bacteria. And the way they do this is they directly inject toxic effectors into neighboring cells. But the way they protect themselves is they have an immunity protein that binds to that toxic effector, so they don't kill themselves and they don't kill their sister cells. So what this is, this big long one here, is that's your immunity protein, and this little one is your effector protein. It's actually the toxin, and they're not sure what the one in between does yet, but it seems to be homologous to the other two, so it does something within that toxic effector story. And what we found was that one of those pathogenic ones without the traditional virulence factors actually had an effector that no one's seen before. So that kind of drew our attention that way. And that's kind of where we ended up publishing on that. And we didn't really know the whole type 6 secretion system story. So it does have this novel type 6 secretion story, but what's the mechanism there? Like, is that enough to cause illness, or is there something else going on? And then recently, and along with that story, trying to figure out if the type 6 secretion system was actually contributing to illness in people, I thought, well, let's look at RNA-seq. So is it being upregulated when you normally see other virulence genes upregulated? And for parahemolyticus, a lot of its virulence genes turn on when it's exposed to bile salt, which makes sense because you eat it, it gets exposed to bile salt, it turns everything on and then gets into your gut. So we did that, and we did actually see the type 6 secretion system being upregulated in the isolates that didn't have any other virulence factors. And then a little earlier this year, someone else published a paper on a shrimp disease called acute pancreatic necrosis disease. And Vibrio, so it's in the environment, infecting other stuff, it actually can infect shrimp and cause this disease. It doesn't use the same virulence factors that it does when it infects humans. It uses PIRA and PIRB. But this paper came out a couple months ago saying that the type 6 secretion system is actually a part of that story as well, the shrimp disease. So you can see in the clinical, it has type 6 secretion 1 and it has type 6 secretion 2. The type 6 secretion system 2 is the one we found that we thought might be related to illness in those other parahemolyticus that didn't have virulence factors. But you'll see for the clinical it has both. For just environmental isolates that didn't cause the shrimp a problem, it doesn't have type 6 secretion system 1. But for the ones that are causing problems in the shrimp, it has both of those type 6 secretion systems, just like the ones that were causing illness in humans without the virulence factors. When they looked at this closer, they were actually seeing the same thing in the shrimp isolates, is that those two genes and the immunity protein were actually changing in the shrimp isolates as well. So that kind of got me thinking, are these guys killing off microbiomes, bacteria, commensal microbes in your gut, and is that actually enough to cause illness? And indeed, the paper published a couple months ago actually did show that when you co-culture things with these two type 6 secretion systems that are able to cause illness in shrimp, you do actually see them killing off commensal organisms in the shrimp. So is that what's also happening with the Vibrio that are causing illness without any traditional virulence factors? Are they coming in and killing off the microbiome? And is just the change in your gut microbes enough to make you sick? I don't actually have an answer to that question, but I think at this point it's actually a pretty good question. Like, is dysbiosis enough for you to have clinical symptoms of an illness? And right when I was going to start answering that question, I came back to Miguel and switched paces again. So hopefully that's something we can get to. But that kind of got me on to this whole interaction. So more and more now, bacterial pathogens are showing that they're actually able to produce different types of effectors and start wiping out your gut microbiome as a precursor to actually causing the illness. But of course, it's not going to wipe out everything in there, so there'll be some bacteria that survive, but how do they select which ones they're going to kill? What's going on in terms of these interactions that allow some to actually survive the attack by the bacterial pathogen, and how often is there a counterattack on the bacterial pathogen, and can we start manipulating this to actually get rid of the bacterial pathogens? So a couple of examples of this, and they're all from 2017, so they're really looking at this now, is Listeria. So the original paper here was published in 2016, but they're publishing more and more on it this year. And what they found is that when certain strains of Listeria infect you, they're able to produce this LLO toxin, and that toxin starts wiping out other bacteria, and that seems to be the only function of that toxin. And not all Listeria have it. Some Listeria are, of course, more virulent than others, and the ones that are particularly virulent are the ones that have this way of fighting the other bacteria. And the story actually gets more interesting than that, is that it's not wiping out all your gut bacteria. It's mainly targeting two different genuses, Alloprobactella and Allobacterium, that are both gram-negative normal colonization guys. And those two genuses are actually big producers of acetic and butyric acid. Acetic and butyric acid are both negative effectors on Listeria. So they can inhibit Listeria growth and activity. So if this is like the whole story, it's kind of mind-blowing that Listeria has a toxin that can specifically wipe out your microbiome that's good at fighting Listeria. So my question is this random and receive stuff that's not there, but is it really that targeted? Does Listeria have the ability to do that? And I don't think we have a full answer to that yet, but it looks like it does, which is kind of mind-blowing. So another story here is that there's been a lot of research on microbiome fighting Vibrio cholera and Vibrio cholera's effect on the microbiome. So this paper was from 2013, and at this time it was known that Vibrio cholera could attack E. coli via this type 6 secretion system. But then a little while later, it was found that certain strains of Pseudomonas can fight back against the Vibrio cholera. So what you'll see in this video is Vibrio cholera expresses a type 6 secretion system that takes out E. coli. But when Vibrio cholera expresses that type 6 secretion system, if your gut microbiome has Pseudomonas aeruginosa, it, in response, expresses a different type 6 secretion system that takes out the Vibrio cholera. So they have the type 6 secretion system labeled here, and you'll see the green ones are the Pseudomonas. When they light up in the little green expressing the type 6 secretion system, some of the Vibrio cholera cells will lyse. And they actually got nice videos of this. Is it good enough to see the Vibrio cholera cells lysing? Yeah, well, the light flashes are the type 6 secretion system being expressed, but the Vibrio cholera cells here, yeah, they lyse. They actually pop. I'll play it again. Did anyone see it? Okay, we're gonna move on. It's there, I swear. This one's a little more obvious. So these guys both have the same immunity protein, so they're going to battle it out with type 6 secretion systems, and this little dot is this cell's type 6 secretion system. But you'll see this cell neighboring it, when it gets attacked by this one, it expresses back and fights back. I swear this one will be easier to see. Okay.
They're secreting something back, like jousting.
Yeah, that's exactly what they're doing. But nobody's winning this one because they all have the same immunity proteins. Yeah, they're jousting, just no one's going to win. So that's actually quite neat because it's a way to talk to your sister cells and not lyse your sister cells, but lyse cells that are attacking you. Pardon?
I can make.
Yeah, exactly. And I have the immunity protein, so you're not going to lyse me. So this is actually kind of interesting from an antibiotic resistance perspective, is that right now with these enteric pathogens, there's a huge antibiotic resistance problem. And this was a couple of years ago, these carbapenems were like the antibiotic of last resort for enteric infections. But they found a carbapenemase, which is the bacterial resistant to that one, on a plasmid. Yeah, which I love your face. Yeah, no, that's not good. So, can we possibly stimulate the microbiome to fight back against these enteric infections? Or could we possibly have some kind of probiotic? You take something that's capable of fighting off whatever enteric pathogen you have and deal with it that way as opposed to dealing with it chemically with antibiotics. So I'm really interested in type 6 secretion systems because of this. And they really only, the first one was discovered, well, the first gene in a type 6 secretion system was discovered in 1996. So we haven't known about them for all that long. And it seems like every year another paper where another bug that has one of these that's capable of fighting bacteria is discovered. And in 2016, Dana actually showed a paper where Salmonella actually has some of these. And Salmonella is actually using these, it looks like, to start fighting your commensal population. It's kind of like Vibrio cholera, but just with the Salmonella. And that's kind of where my lab is going in the next little while. So when I first came to McGill, there's a Salmonella sequencing project going on right now where they're sequencing 5,000 strains of Salmonella, and they're actually trying to get information on those 5,000 strains of how virulent they are in the mouse model and correlate those two data sets, which is a huge undertaking, but it's going to be an amazing data set when they're finished. So I thought I'd kind of piggyback on this study and it's like, well, feed a couple mice Salmonella and let's see what it does to their microbiome. Easy peasy. And they did that for me. But what we saw was that absolutely nothing changed, which was not what I was expecting. Like, these mice were actually pretty sick, and their microbiome wasn't changing all that much. INV, and SSE are two important virulence factors in Salmonella. So even when you knock out the virulence factors, nothing changes. So it's not just a virulence thing, at least with this strain. Probably is a virulence thing with some strains, but not this one. So that got me thinking, well, let's look at type 6 secretion systems. What do we know about type 6 secretion systems in Salmonella? And the answer is not actually that much, which is kind of surprising to me. But Salmonella enterica actually has pretty complex phylogenetics. They're all divided up into these serovars. So beyond the species level, there's different subtypings of Salmonella, and each one seems to do a slightly different thing. So typhi causes typhoid fever, which is actually a pretty serious problem for humans. Typhimurium causes gastroenteritis in humans, but if you get down to something like Dublin, not such a problem for humans, but Dublin's a problem for cows. Heidelberg's a problem for people, and I'm looking for one that's a problem with chickens.
Gallinarum.
Thank you. Gallinarums are problems for chickens. But in terms of type 6 secretion systems, SPI6 is one, and you see that in a wide variety of different Salmonellas. SPI19, you see that in more of the animal ones, although enteritis goes into humans too. SPI20 and 21, they're more rare. But the thing is that when we looked at the genomes to see how many different type 6 secretion systems were all Salmonella, the last time someone did that was in 2009 when there were 44 whole genome sequences available. Does anyone know how many Salmonella? Yeah.
Now, since then.
Yeah, like today.
Well, soon to be 5,000.
Yeah.
But
If you combine that with with the NCBI database, now there's almost 10,000. So it's time to go looking for these things again. So I have a master's project hopefully coming up in 2018 where someone's actually going to go do that. I did a quick search just like with my limited programming abilities one evening for fun. And I actually found what I think are 3 new ones. So I think there's a lot of them there. So hopefully we'll get into that soon. Cold culture assays too. So they've done that for the SPI6, but no one's actually done that for the other 3 type 6 secretion systems. So are they actually fighting other bacterial pathogens or other bacteria, or are they doing something totally different? And the big question is, how are other bacteria responding to this? Is it going to be like the E. coli, Pseudomonas, Vibrio cholera, where you do actually get different species that are able to fight back, or is it just going to be an obliteration thing? So that SPI6, which is a little more common among the bugs, Sana did look at that, and it was actually interesting because that particular type 6 secretion system is very easily able to kill K. oxytosa, K. varicola, and the E. coli K12, so like the lab strain of E. coli. But it's not actually able to kill some other strains of commensals. So it seems to be selectively killing bacteria in your gut, but we don't know what the selective criteria is yet. And I think, provided I get some funding for it, that's going to be a PhD project coming up to figure out what it's doing to the total population, not just a couple different strains of commensals. In addition, how does your gut microbiota respond to a salt with that type 6 secretion system? And it would be really cool to look at this using transcriptomics or metagenomics as well. And then just another kind of little side project we're planning is that we don't really want to be tied down to Salmonella or a story like that. We're going to start looking at a couple other bacterial pathogens as well. And Staphylococcus aureus is actually one of my favorite foodborne pathogens that no one thinks of as a foodborne pathogen, but it's caused some pretty interesting outbreaks. But it also causes mastitis in cows. And the interesting thing is if we go start sequencing raw milk from individual cows, you can figure out which ones have had mastitis in the past because their microbiomes are actually quite different. So MQ is cows that have had mastitis, HQ is cows that haven't had mastitis, and they're actually, like, for the rest of the life of that cow, you can tell that they've had mastitis based on these changes in their microbiome. And once a cow has had mastitis, it's very likely to get it a second time and a third time. And when that happens, most people get rid of the cow from their herd, which is, it's sad, but it's also a problem in productivity for the dairy industry. So if you could figure out these problems, you might be able to alter the microbiome in a way to prevent future Staph aureus infections. So that was one thing we were going to look at, as well as taking a look at what happens pre-infection. So in a normal microbiome, in a cow that's never had staph before, is there something happening to it before it becomes infected that makes it more prone to infection, is actually the question we're going to be addressing. So in any given year, 15% of a dairy herd will actually get mastitis. So we have 70 cows out at McDonald campus. We're just going to take milk from them every day and wait for one of them to get mastitis and then go back and start sequencing the microbiome. So this project did actually get funded. So we have a master's student position open to do that from 2018. So why do we care? Hopefully the idea here is that you could actually alter the microbiome to start preventing infection or maybe even cure infection. And the question I always get from people who aren't microbiome people is, can you actually do that? And there have been a couple of studies that actually show you can do that. It's actually very effective. So I think this paper is actually 10 weeks out. So these are people who had Clostridium difficile infections, which is a post-antibiotic. I have no microbiome left, so I get Clostridium difficile. And you can see their microbiomes are not things that you normally find in a healthy microbiome. It's full of Klebsiella and Enterobacter and Citrobacter, whereas the donors have a healthier population with Enterococcus and Firmicutes, whatever he is. But 6 weeks out, this donor went into this guy and so forth, back and forth. Sort of 10 weeks out, you actually see a microbiome more like the donor. So it is actually taking and staying taken and not going back to the way it was. So there's some evidence this will work. And this slide has less to do with it, but it's looking at antibiotic resistance genes. So after a nice bout of antimicrobial treatment, and before someone gets, or after they've gotten Clostridium difficile, you'll see the donors don't have any genes in their microbiome that are associated with antibiotic resistance, whereas the people who have just completed the course of antibiotics actually do have a lot of genes that are associated with that. When you give the donor the when you give the person the fecal transplant from the donor, you actually see a reversion back to the have any antibiotic resistance anymore. So it's kind of interesting, and it's just another piece of evidence that these microbiome transplants do seem to work. And that's it for today.